Hi, my name is Jeremy Meter, and I'm going to be discussing mRNA modifications. So after transcription, an mRNA molecule is produced. However, it is not ready to be exported to the nucleus just yet. If it is exported like this, without modifications, what will happen is that the 5' and 3' ends will be degraded by ribonucleases. And if it makes it to the ribosome, introns will be expressed, which are non-coding regions, which will affect how the gene is expressed. So, in order to prevent this, there are 4 different mechanisms that RNA has to undergo to be exported to the nucleus for proper translation by the ribosomes. The first one is known as 5' addition of 7-methylguanine. And what happens is that with the help of a capping enzyme, it facilitates the reaction between a 7-methylguanine complex and the primary mRNA transcript, and it forms a triphosphate bridge. Once this happens, the cap will actually serve as the ribosome binding site and protect the 5' end from being degraded by ribonucleases. The second mechanism that mRNA undergoes is the addition of a poly A tail by poly A polymerase. Now in this one, there is going to be an overtranscripted region at the 3' end, but there is a sequence that is going to be recognized by poly A polymerase, and it's AAUAAA. 11 to 30 nucleotides downstream from that will be a cleaving site. This is where the poly A polymerase will cleave the overtranscripted site. So it'll leave this segment plus 11 to 30 nucleotides depending on where the cleaving site actually is in this area. After it cleaves, the addition of poly A will be catalyzed by the poly A polymerase once again, and this will be added after the cleaving site. So as you see here, now our mRNA has a 5' cap and the poly A tail. And the poly A tail is going to prevent the 3' end from being degraded by ribonucleases, and is also going to be needed for full initiation of translation. Now, the third mechanism that we are going to discuss is going to be mRNA splicing. During mRNA splicing, the regions known as introns are going to be cut out. They're going to be cut out with something called the spliceosome. The spliceosome is going to recognize these GU at the 5' ends and AG sequences at the 3' ends. Now, the GU and AG are going to be called the splice sites, and that is where the intron is going to be cut out and the exons will be spliced together. After the introns are spliced, the only thing left is the exons, which is going to be our gene. So now our mRNA has a G' cap without introns, only exons, and a poly A tail at the 3' end. And the mRNA splicing is going to be needed for the variation in protein. So if there's alternative splicing, then the gene can actually express different forms of the protein. The fourth mechanism and the last mechanism is mRNA editing. Now, mRNA editing is going to just be a change, deletion, or addition of a nucleotide sequence to the mRNA transcript to alter an amino acid. The purpose of this is to be able to create variation in proteins. After it's gone through these 4 mechanisms of modification to the mRNA, it's known as mature RNA, and it'll actually be ready to be exported from the nucleus to a ribosome and be translated as a protein.